All right, so I wanted to do a short lecture here to finish up our discussion about genetics and relationship to schizophrenia. And I wanted to introduce the concept of pruning. Okay, and pruning is basically what it sounds like, except what we're pruning here are the neurites, okay, and neural connections and synapses at formation. So remember, so far there's at least my knowledge, I think probably grows by the day, about 130 genes identified with schizophrenia. Okay, so we're not talking about things. And again, that's one of the reasons that things like these are spectrums. Okay, we've got, and they, what one pharmaceutical agent that will work for one individual won't necessarily work for another, or it takes different levels or whatever, because we have so many different things that can play a role. But one of them that is understood, and not only affects schizophrenia, but it helps us think about some other genetically related issues, is synaptic pruning. Okay, and particularly with schizophrenia, as you'll see, it's overexpression. There's also underexpression of pruning that plays a role in some of these as well. But basically, we just kind of look at synapse formation. So, brain density, right? How much connections we have. And early on, during gestation, we start to have neurite connections in brain development. A newborn has some amount, and it differs among individuals. And these tend to increase fairly rapidly over time. These connections build through the first couple of years of our lives until we reach about 2 to 3 years old, which is about as dense as they're going to get. Okay, and we have lots of neural connections at that point. It's one of the reasons that potentially things like little kids sometimes complain that the crust of bread tastes terrible. Well, they may have more neural connections than when they're 10 or 12, or now as adults, we go, oh, crust of bread tastes good. There's all sorts of connections there that are out. Well, then once we get past this couple of years, 2 to 3 years postpartum fruition time, we start to prune. And we spend a number of years pruning out and making those connections as speech develops, as our motor skills develop, etc. We start keeping those connections that seem to matter, and reducing those connections that tend to not play a role. So the density there and becomes more focused in things. And this is one of those interesting areas. Now this continues on up and well into our teens. There are lots of genes, as you can expect, that have to do with pruning, right? But one of them that I wanted to look at, just as part of this, was a gene found on chromosome number 6. This is chromosome number 6. It's up here in the long arm, or the short arm, and the top up here. This is the C4A gene and C4B gene. This was also two variants of this gene, but they're just kind of indicate where they are up in chromosome 6. So what do these genes do? Well, it's actually, when I say the gene, it's actually a single gene that's transcribed, and then there's post-translational modification, major modification of that gene relative to expression. Okay, and meaning that we cut out there's a lot of pruning of the gene, of the transcript itself, post-transcriptional. To make it either a short variant, which is known as which contains the exons that make it the C4B variant, which is one of the common ones. There's the C4A long, C4B short. There's long, etc. And there's all these variants of this. And there's now you're saying, well, how does it get C4A long, because this is one of those situations where you have different, you have sort of a co-mingling of the genes by RNA before you get translation, right? And so, we've looked at that before, and you certainly talked about it in genetics and molecular genetics. The fact that these transcripts, you get two different transcripts, and very often those will combine in some form to make a new gene. That's the reason we only have about 19,000 known functional protein genes, but we have hundreds of thousands of proteins, right? It's because we can do heterogenes and homogeneous conations. Now, all these produce the C4 protein, and it depends on the variant. Obviously, there's different variation in what that protein looks like and how it's going to function. And with these greater links here, you get an increased relationship to schizophrenia. And we think that works, okay? One of the ways we think that works has to do with some of this pruning activity, okay? So we've got our dendrite here, we've got our C4 genes being produced, depends on which ones you get. They're carried out through on the axon, so they're, kinesin is transporting them out along the pathways here. And then they are utilized at the synapses for pruning, okay? And if you have the long ones, they tend to do more pruning, and the real short ones tend to do less pruning, okay? That's basically how it works. So depending on the variants that you have, and what can cause this variation here can be either the regulatory control region, enhancer region type regions, or there can actually be, turns out there's variation in the maturing of the message. So those proteins that are involved in splicing and things can actually cause relationships. So some individuals are because of the splicing variants, others are because of transcriptional variants and things. But anyway, we get an increased rate of synaptic pruning and increased risk of schizophrenia with over-pruning, overexpression. So if you get too much expression of this, which is going to happen here, and then when both are there, you're going to get much more production. Okay, so if we look at this as a general, you're in the middle is schizophrenia. Okay, schizophrenia is excessive pruning. Right, there's not enough connections being made. We've cut out too much of the connectivities, and we've broken down some of the normal pathways that things tend to take. So things tend to go across the same pathway for different things. And the microglia cells, for example, are one. And we get all sorts of other, you can look up, NOS, activation and all these sorts of things that can occur. Now, the reason I wanted to talk about this pruning, besides the fact that I just want to introduce the idea that it happens and that it's under genetic control, is that you can have other things happen. There's cases where you can have reduced pruning. Okay, you have way too many connections that remain once we reach adolescence. This is one of the things that is related to autism. And there are all sorts of expressional things that can go on. This can also be due with, methylation. So there's epigenetic things that go on. It's one of those things that makes things like the autism a spectrum, right? Now, some kids are autistic; they're just a little bit more active than others. Some kids are just, basically are dysfunctional as far as their ability to reason and do things in a normal, cohesive way. So the disorder runs a whole, it's a spectrum, and part of that has to do with things like methylation of genes, et cetera, and alterations. But what we do see in a lot of the core types of autistic outcomes is that there is reduced pruning. These individuals have a lot of connections that go on, and those connections are firing too much, and they're not pruned down to where they're centralized and in the particular brain regions that they need to be. On the complete other end of this, you get a more complex things like temporal lobe epilepsy, where is a result of both outcomes. Okay, there's reduced pruning early. Okay, so these individuals don't prune up, so much like you would see in an autistic outcome, and then somehow the system kicks in and does too much pruning. Okay, and so you get these epileptic things. The system basically overloads as one of the things. This is primarily an autosomal dominant system. We could talk, again, like I said, on every topic we talk about in here in genetics, we could talk about each one of these over and over, excess, what's really happening, why is, phagocytosis going on here, what's the, what's really going on here, how this chemotaxis occurs, what chemicals are involved, what's causing the epileptic event to occur due to a buildup. So we could talk about all these. I just wanted to point out that pruning exists, okay? and that it's under genetic control again, and that you can either have too much, too little, or you can have some combination, and these lead to different, but these are all associated outcomes from essentially from pruning, okay? Even though completely different gene systems may be involved, completely different outcomes, forms, they can all relate to a common form. Okay, so that basically concludes that. I want to talk a little bit about dementia and some of the various forms of what we would consider under the spectrum of dementia on the next lecture, and then we'll get into some of the other forms as well.